All right, so this will be our last video for this week. So when we think about mutations, we've kind of summarized, it can help, not help, whatever. But we do know that mutations are the reason why we have acquired drug resistance. The most famous acquired drug resistance is in the case of antibiotic-resistant bacteria, which we've known about this for over 20 years. So I'm not going to go into the story about that. I hope that you do know it, but essentially microorganisms that are not killed by a drug. So if they're able to survive, if it's ciprofloxacin or Cipro or amoxicillin, or maybe it's even, some other kind of medication. But if a bacterial cell is not killed by that antibiotic, then essentially it can live, survive, reproduce, and have more of itself or offspring that also have that mutation and so have that drug resistance. So essentially we have been running what they call a co-evolutionary arms race with essentially microorganisms, disease-causing microorganisms, because we use antibiotics and at some point in time they become ineffective. Oftentimes this is within individuals, okay? So if you've been on antibiotics many, many times during your lifetime, it may now be that you need a combination of antibiotics, or you need one of our kind of big guns antibiotics to treat maybe it's just a sinus infection. So it's important to know that taking antibiotics, using antibacterial chemicals can actually favor those organisms that have the mutation that confer resistance to those medications or to those chemicals. My next slide is just going to go back to thinking about proteins. So you know that DNA codes for RNA through transcription. RNA then is going to code for a protein through translation. So you know that. And so we just want to take that idea, I don't know what's going on here. Anyway, take that idea of a mutation and think about how does that affect a protein. So the mutation may not change the protein at all. The structure and the function, no change, right? So no change in the functioning of the organism. So you might also have the situation where you have some or complete loss of the normal protein structure or function. So it may or may not have a negative effect on the organism. And then the last situation is you may have some improvement in protein functioning, but it may or may not have a beneficial effect on an organism. So just because you have the mutation, it may not change the protein. It could change the protein in a positive or negative way, but it may or may not have any real life beneficial or negative effect for that organism. So looping back around to the big differences between DNA and RNA, and I want to think about DNA viruses and RNA viruses. So DNA viruses, which you don't really talk so much about in this lecture because they're pretty straightforward in how they're using, the DNA replication machinery of their host cell. But essentially our DNA viruses are much larger and more stable than our RNA viruses. They also essentially affect the nucleus or the area of the nuclear material. So that's where they're housed, and so they're somewhat protected there, especially in eukaryotic cells that have a nuclear membrane. But our DNA viruses include things like adenovirus, herpes simplex virus, and human papillomavirus. So they're very, very stable. But when we take a look at looking at the differences between DNA and RNA, one of the biggest differences is the fact that deoxyribose is more chemically stable because of this single hydrogen atom here on the sugar, whereas ribose has the hydroxy group. Okay, so let's move on to examples of RNA viruses, and I'm going to tell you that they are unstable. They tend to have a smaller genome. They tend to hijack the machinery of their host cells out in the cytoplasm, again, using those ribosomes and genetic codes associated with RNA to make more RNA. So these particular viruses are a little bit harder to treat with vaccines, simply because they are chemically unstable. It may just take one mutation or one change, and now they are immune, if you will, to our vaccines. So you might have remembered during the height of the pandemic with the coronavirus or COVID-19 that essentially they were coming out with vaccines and another version of the vaccine, and another version of the vaccine. I think I had like 8 doses of vaccines off and on during those years. I was in nursing school and then practicing nursing, and so it was super important to get the newest, latest, greatest vaccines. But essentially, what we know is that if the circulating virus, so in this case, the coronavirus, has a mutation, then when you contract it, your immune response through memory cells may not even recognize that as a coronavirus. So you didn't attack that with essentially your B cells. So then you come down with the illness and then you can spread it. So our coronaviruses are pretty unstable. And because of that, we have needed to be careful with those viruses. It also makes them very, very contagious. And of course, they're mutating rapidly. And so it can spread through a population very quickly. So those are all big problems. Now, let's think about the flu virus. So if you get a flu vaccine, essentially what they're doing is they're taking a look at what new viruses are starting to appear on the global scale. And oftentimes you're looking towards China and essentially taking note of what appears to be the most prevalent flus at the time. And then they began manufacturing our fall flu vaccines, incorporating those particular strains of influenza. So it's a little bit of a crapshoot and a cocktail. So you try your best, or they try their best to predict what it's going to be. And then the cocktail is you're getting about 3 different immunity to 3 different viruses that might be part of our regular flu season. So, for example, I don't know if you all remember, I believe it was 2009 when we had a situation where we had another pandemic flu. I believe they were calling it H5N1, and essentially it only needed one specific mutation to become easily transmitted between humans, and so they came out with a vaccine. Basically, by the time they came out with the vaccine, it was not effective, but everybody felt better that they had the vaccine and moved on. So some other RNA viruses, dengue fever, West Nile virus, Ebola. And then I did want to make note of measles. So the measles vaccine works really well because you would have to evolve, or there'd have to be 5 different mutations for that particular variant to go unseen by your immune system. So that's why it works really well. And you don't need to have repeated doses of the measles vaccine like you would the seasonal flu. HIV is here. Of course, we know it's a retrovirus. So let's talk a little bit about vertical and horizontal gene transfer. So when you think of vertical, up and down, this is simply the regular way we're thinking about reproduction and passing on heritable traits. So you have your original mother cell, it takes its DNA, copies it, and makes 2 daughter cells. So you're going from one generation to the next, to the next, to the next. When we think about horizontal transfer of genes, we're thinking about essentially the transmission of genetic material at the same generation. So you don't need to know these 3 words: transduction, conjugation, transformation, but you can see essentially under transduction, you have a bacteriophage. Remember, this is a virus that infects bacteria. And once inside that virus, it is picking up a piece of genetic material from the plasmid, or not from the plasmid, but from the circular DNA from that bacterial cell. So now it's picking that up, and then it can essentially infect other cells by just doing what it did to the original cell. Okay. In conjugation, this is the closest thing to what we call bacterial sex. But the funny thing about it is, generally speaking, you're going from one cell to another cell. So again, this is an example of horizontal transfer. So in this case, you have the original donor, and it's got a piece of genetic material. In this case, it's showing us the plasmid, and that plasmid then is offering up this material to the other cell. Okay, so this again is not involving entire replication of the genome from one parent to essentially a parent cell into the daughter cells. In the last column, we see transformation. So essentially, you've got this particular genetic material inside a bacterial cell, and the bacterium dies, and now you have this genetic material that is picked up by another bacterium. So again, we're not thinking of this horizontal, of this vertical transfer from the parent cell to the daughter cells, but instead across cells of the same generation. All right, moving to chapter 19, I want to talk about antigenic shift and antigenic drift. And this is going to help us understand the difference between like seasonal flus and pandemic type flus. Okay, so when you look at the word drift, when I think about drift, I think about things just moving subtly and slowly, kind of one direction or another. So in figure A on the left-hand side, it is showing us a virus A, and that virus A is going to undergo some mutations, and essentially what appears to be happening when we see the virus B is that one of the two antigens on the outside of the cell now looks a little bit different. So the mutated hemagglutinin has a little bit more of a kind of flower appearance to it, as opposed to like that green tree on virus A. But the other particular antigen is exactly the same. So, essentially what we have here is what we call point mutation. So you have one single change that essentially just changed one of those antigens a bit. Okay, so it's a little bit different, but it's not like drastically different. But essentially, when we think about seasonal flus, we know that we're still going to have a new flu vaccine because our immune systems won't recognize that. In figure B, we see antigenic shift. So this is where you have a pretty radical change in the organism such that we have a new virus. So you got virus A and virus B, and essentially what they have done is combine their genetic material. And so the resulting virus C has some properties from virus A and some properties from virus B, but makes them different from both of those. So this is a real serious situation because we're picking up genetic material, and these particular viruses can change very quickly, and even our herd immunity is not going to matter because it's so different that our bodies don't recognize them. Okay, so let's take a look at a different figure. This is not in your textbook, but I thought it was helpful. So essentially, there are several influenzas that are easily transferred between various vertebrate animals. And so in this figure, we see that there is a bird flu or avian flu, and then certainly a human flu. We've recognized them by their H and N's. And so each of these can actually be picked up by a pig. And in the pig, then we see that we are taking those two viruses and making a new novel virus. Okay, that virus here has been named H3N2, and it is a pandemic type flu. So easily contagious. It's easily harmful to humans because we don't have any particular immunity to that virus. So you see that is an example of antigenic shift. So there's this genetic reassortment, and we have this novel virus as a result. In the lower figure, we can just see, okay, well, this is our normal seasonal flus, and we have a slight alteration, maybe just a point mutation, causing one of those antigens to be slightly different. But essentially, we may or may not have much resistance to resistance to that particular virus. Let me just look at my notes to see if there's something else I wanted to say. What we do know is that those viruses resulting from antigenic shift are very, very concerning because they spread like crazy. And we don't have the antibodies to even recognize them. Okay, y'all. I think that's it. Please let me know if you have questions, and we'll see you next week.